welcome. I'm delighted this afternoon, by 5-7 seconds, to welcome again to ASU and to introduce to you very briefly Dr. Sharon DeWitte. She's currently an associate professor in anthropology and biological sciences at the University of South Carolina, where she moved up the ranks quickly since she joined the faculty there in 2011. She previously was an assistant professor of anthropology at the State University of New York in Albany, beginning in 2006. I've known Sharon since she was a scary, smart undergraduate. She came to Campsville in our bioarchaeology program in 1998, when she was just about to graduate from Sonoma College in California. And she received her BA there in the next year, graduating magna cum laude. She disappeared from the scene, most graduate students do for a while, and then popped up again as a graduate student who was publishing before her dissertation, but on her important dissertation work. And she received her PhD then from the Pennsylvania State University in 2006. In 2008, she published a very important, she was a senior author, important article in PNAS on the Black Death and selected mortality and frailty, a topic she's extended and expanded and taken in some very exciting new ways, the virtual explosion of publications since then. And she'll be talking about that subject today. One of the aspects of her talk today will be developed from a recent 2014 PLOS One article, which I just saw has been listed as one of the top 20 articles in terms of field influence, public influence by PLOS One. So I think that speaks well of the visibility and applicability and the significance of her work. With that in mind, I'll speak no more and turn the podium over to Sharon. Please join me in welcoming her.
Thank you, Jane, for that wonderful introduction. Thank you to Dr. Nessie and to the Center for having me here today. And thanks to all of you for coming out to listen to my talk. So as Jane mentioned, I'm going to be talking today about my research on epidemic diseases in the past, and specifically on the medieval Black Death, and what this might tell us about current and future threats to human populations. So if we can use Time magazine as our sort of standard for what's considered important. Time magazine did select the Ebola fighters as their persons or people of the year for 2014. And this just underlines how strongly the current Ebola epidemic, which began in December of 2013, how much that has focused the world's attention, not just on this particular emerging disease, but on emerging diseases in general. Emerging diseases are those that are new to human populations or that are increasing in incidence or in geographic distribution. Dozens of diseases have emerged in the past few decades. Those include HIV, SARS, West Nile virus, and of course Ebola. And new diseases are certain to emerge in the future given human encroachment upon environments and the speed and scale of global transportation, the overuse of antibiotics, which is leading to the reemergence of certain diseases like tuberculosis, and other factors. In the case of Ebola, the previous outbreaks of the epidemic were incredibly frightening in terms of case fatality rates, which were as high as 90% in some outbreaks. But what is so terrifying about the current outbreak is the unprecedented number of victims and also the very large geographic distribution. According to the Centers for Disease Control and Prevention, as of Sunday, there have been, in just the countries of Guinea, Sierra Leone, and Liberia, over 21,000 reported cases of Ebola and over 8,600 reported deaths from Ebola. So this makes it by far the largest Ebola epidemic in human history. The previous outbreaks of Ebola affected at most a few hundred individuals and were mostly restricted to forested regions. According to the World Health Organization, the current outbreak of Ebola does not differ from previous outbreaks in terms of the clinical course of the disease or transmissibility or case fatality rates or other characteristics. So the disease itself in the current outbreak doesn't differ from previous outbreaks. What is different, according to the World Health Organization, is the human context of the disease, so attributes of the human population that is affected now. And that includes large intermixing populations in which there's very easy transport between rural areas and dense urban centers, and there have also been insufficient control measures. What's important to note about the current epidemic is that the scale of it was not inevitable. Ebola was introduced into dense urban centers in Nigeria, but in that country there have only been 20 reported cases and only 8 deaths. And that's because very rigorous control measures were swiftly implemented once the disease was introduced. So Ebola is a very dramatic example of a contemporary emerging disease, and it's obviously been receiving a lot of attention. Emerging disease research primarily focuses on understanding when, where, and why new diseases will emerge, and in trying to control their emergence once they actually do emerge. So most of the research that's done in emerging diseases has revealed the factors that favor emergence, epidemiological patterns of contemporary diseases, mortality patterns in particular. But there remain very important questions about emerging diseases in general. That includes who is at the highest risk of disease and death during emerging disease epidemics. How do epidemiological patterns change following emergence? One of the things that might be happening with Ebola, according to the World Health Organization, is that it could become endemic to human populations, and that's something that hasn't before been considered a possibility. We also want to know more about what is the nature of the interactions between or among emerging diseases, social, political, economic, environmental conditions, and human biology. What are the long-term and also the shorter-term effects on human health and demography? And how do the pathogens that cause emerging diseases co-evolve with humans and also non-human hosts? And these questions persist in large part because most of the emerging disease research that's being done and has been done over the past few decades has focused on current emerging diseases. And that's clearly understandable because these are the diseases that are killing people on the ground today and have the potential to kill people in the future. However, this focus on recent emerging diseases means that we really do lack the temporal depth that's necessary to fully examine changes that can occur in the behavior of emerging diseases and also the long-term interactions between emerging diseases and their human hosts. And without a complete understanding of these phenomena, our ability to actually respond to or perhaps even prevent disease emergence is hindered. I don't want to say the good news, but maybe the good news is emerging diseases are not just a recent phenomenon. They were important in the past. And the reason that that's potentially good news is it means that if we can look at emerging diseases in the past, that might give us some tools for dealing more effectively with emerging diseases today and also in the future. For over 10 years, I have focused my research primarily on the medieval Black Death. And the dates that I'm showing here, 1347, 1351, those are the dates during which the Black Death was at its height in Europe. The Black Death did affect a much larger region of the Old World. Scholarship is increasingly focusing beyond Europe, but a lot of what we know and what I'm going to be talking about today focuses on Europe. So the research that I've done on the Black Death, what I do is I use paleodemographic and paleoepidemiological approaches. So my data are coming from human skeletal samples, and I'm primarily interested in population-level trends in mortality, survivorship, and health. I do take the perspective that the Black Death was an emerging disease of the 14th century. We do have genetic evidence that the Black Death was caused by the same pathogen that caused the 6th century plague of Justinian, and also causes the modern bubonic plague, so that's the bacterium Yersinia pestis. So strictly speaking, the Black Death, that disease was not new to humans in general, but it was clearly new to medieval populations of the Old World. The Black Death had extraordinarily high mortality levels, killed an estimated 30 to perhaps 60% of affected populations. Some populations suffered even higher losses. And by leading to such dramatic depopulation, the Black Death led to, or at least accelerated, really important changes, not just demographically, but also socially, economically, and politically. And because of that, the Black Death has fascinated people, not just from a variety of academic disciplines, but just the general public. And like a lot of people, I am interested in the Black Death. And I am interested in the Black Death because I am a biological anthropologist, and ultimately what I'm most interested in is human evolution and human biological variation and the things that can shape both of those. Infectious diseases can play a very powerful role in driving human evolution and also shaping human biological variation. So it's really important to understand them and how they've behaved in the past. I'm also, as an anthropologist in general, very interested in the biological, social, political, economic factors that shape risks of disease and mortality under a variety of conditions, both catastrophic conditions like the Black Death and also under normal mortality conditions. I'm also very interested in how the human context of a disease can shape the epidemiology of the disease and also have a role in the evolution of that disease. So I'm not interested just in how diseases affect humans, but also how humans can shape the disease itself. The Black Death provides an amazing opportunity to examine all of these various issues. So I'm referring to the Black Death as an excellent case study for examining emerging disease in the past, and that's for several reasons. For one, the Black Death was a massive epidemic. It was also incredibly short-lived, and it was very well dated. That means we know when and where the Black Death happened, and because of that, we can examine the environmental, demographic, economic, social, political conditions that existed at the time of the Black Death. That means that we have the opportunity to examine factors that might have favored the emergence of the Black Death, and we can also examine how the Black Death might have shaped some of those factors. We also have cemeteries that were used exclusively during the Black Death. That means that we have large samples, large from an archaeological perspective. We have large samples of people who died within a very, very short period of time from a single known cause of death. This is really unusual for bioarchaeology. And one of the things that means is that we don't have to rely on skeletal lesions in order to identify victims of this particular disease. And that's good because as informative as skeletal lesions can be, they also can be very problematic. And in general, skeletal lesions tend to have low specificity and low sensitivity. So what that means is that lots of different diseases or disorders can lead to similar, if not identical, skeletal lesions, so low specificity. And also, individuals who have a disorder or a disease that could lead to a skeletal lesion don't all necessarily develop that skeletal lesion, so there's low sensitivity. But with these Black Death cemeteries, we don't have to rely on skeletal lesions to diagnose victims. In addition to exclusive Black Death cemeteries, there are also exclusive pre- and post-Black Death assemblages from places like London, where I work, Denmark, Germany. And what that means is that we can approximate a real-time examination of the effects that the Black Death had on a variety of conditions. So we can look at the effects of the Black Death on demographics, on health, on human genetic variation. That means that we can look at things like whether the Black Death might have acted as a very strong force of natural selection and drove up the frequencies of variants associated with disease susceptibility or immune competence. Or we can look at whether the Black Death might have led to genetic drift and in that case resulted in substantial but more random changes in human genetic variation. And then lastly, the Black Death was caused, as I mentioned, by the same bacterium that causes modern bubonic plague. This is a disease that still kills hundreds of people every year. It's primarily a disease of non-human animals, and strictly speaking, it's not really a human disease. It affects humans, but we are not the primary hosts. And because of that, it's impossible to eradicate the disease. Some people are afraid that it has the potential to reemerge as an incredibly dangerous disease for modern humans. The Black Death pathogen, Yersinia pestis, also caused, as I mentioned, the plague of Justinian, which was in the 6th century. This means that we're looking at a disease that has affected humans for at least 1,500 years, and that gives us the opportunity to examine trends in a disease over a fairly long period of time and look at how humans might have changed in response to that disease and how the disease might have changed as well. The work that I've done previously with the Black Death focused on a single assemblage. It's the East Smithfield Cemetery from London. This is a photograph taken during excavation of East Smithfield in the 1980s. This cemetery is one of just a very tiny number of large excavated cemeteries in Europe that has both excellent documentary and archaeological evidence linking it to the Black Death, the mid-14th century epidemic. East Smithfield is located, it was established on agricultural land just outside the city walls at the eastern end of the city, literally across the street from the Tower of London. And here's the North Tower of Tower Bridge. If you've been to London Tower Station, the tube station's right there. East Smithfield was established before the Black Death even arrived in London. So people in the city knew the disease was coming. They set aside land in advance of the epidemic's arrival. And then the epidemic arrived, and East Smithfield was only used during the epidemic between 1349 and 1350. Everyone who's buried in East Smithfield died during the Black Death, and that means most, if not all, the people buried there were victims of the Black Death. And it provides this really amazing opportunity to examine mortality patterns of the Black Death empirically. And what I was interested in previously, and this is something that drives a lot of my research, was in examining variation in risk of mortality, in this case during the Black Death. So in general, I'm interested in the phenomena of heterogeneous frailty and selective mortality. And in the case of the Black Death, I was interested in testing long-held assumptions that the Black Death killed indiscriminately. Most normal non-epidemic causes of death tend to be selective. They tend to target the frailest, weakest individuals within a population. But previously it wasn't clear whether the Black Death did that as well or if it killed indiscriminately. And so I used a sample of nearly 500 individuals from East Smithfield to examine variation in mortality empirically. What I did was I compared this sample from the Black Death Cemetery to a large pre-Black Death normal mortality sample. So a sample of people who died before the Black Death under normal mortality conditions. And I used that normal mortality sample as close to a control sample as I could get to see how the Black Death patterns might have varied from normal conditions. And these are the three major patterns that I focused on. So I was interested in selectivity of the Black Death with respect to sex. So whether males or females were at higher risk than the other, or if both sexes faced roughly equal risks. This is important not just in terms of understanding how the Black Death behaved, but also in terms of understanding the antiquity of modern sex differentials in morbidity and mortality. So this is something I'm interested in under a variety of conditions. I was also interested in determining whether the Black Death was selective with respect to pre-existing health condition, or more simply put, frailty. So in the case of the Black Death, I was interested in determining if individuals who are already in relatively poor health because of exposure to disease or because of malnutrition or other physiological stressors, if those individuals were thereafter more likely to die during the Black Death than their peers who'd been healthy up to the point of the Black Death. And then lastly, I was interested in whether there was variation in risk of death during the epidemic across the lifespan. What I did in this research and what my general approach is the application of hazard models. And hazard models are beneficial for bioarchaeological analyses because they allow for the estimation of mortality patterns without potentially imposing incorrect patterns on skeletal data. So with hazard models, I fit a parametric model of mortality to data from a cemetery, and then I can estimate various covariates, like a sex covariate or a social status covariate or other types of covariates. With hazard models, the ones that we use in bioarchaeology, they typically have relatively few parameters. So some of the models I use have 5 parameters, maybe 6 or 7 at the most. Because these models have relatively few parameters, they can be applied to the typically small samples that we get from archaeological sites. These models can accommodate missing data, and they can smooth the random variation that exists in small sample sizes without imposing potentially incorrect patterns on those data. So they're a very powerful way to approach the fairly flawed data that we have from archaeological sites. This is just one example of a hazard model that I've used, and I used it for my Black Death research. And this is a model that allowed me to examine whether the Black Death was selective with respect to frailty. This is a model that was developed specifically for paleodemographic use by Usher. It's a 3-state model. The third state is death. This is what the state everybody is in. That's in a cemetery. So everyone I look at is dead. And then the 2 living states are the states that an individual could have been in just prior to the transition to death. State 1 includes individuals who lack skeletal lesions or stress markers, and state 2 includes those who have skeletal lesions or stress markers. The stress markers that I use in my research include things like relatively short adult stature, dental defects, lesions that are commonly attributed to anemia, lesions that are associated with trauma or infectious processes. Those of you who are interested in the specific lesions, I'm happy to talk about those more in the question and answer session. But for now, what this model allows me to do is estimate the difference in risk of death between people who have these visible stress markers and their age peers who lack them. So what I do with this model is I specify a baseline hazard from state 1. The hazard that I use will depend on the composition of my sample. If I'm including individuals of all ages, I'll use the Seiler model, which I'm showing here. The Seiler model is a 5-parameter parsimonious model of mortality across the lifespan. It's been shown repeatedly to fit almost every known human pattern of mortality, and it describes the so-called bathtub curve of mortality, where mortality risk is highest at birth, it declines, and then increases again during senescent years. If I'm looking just at adults, I'll use a simpler model that just describes the adult component or senescent component of mortality. So I specify a baseline hazard, and then this K2 parameter of the model indicates whether individuals who have a particular stress marker were at elevated or potentially reduced risks of mortality compared to their age peers who lacked that stress marker. And this is an important feature of the model because I don't constrain the value that that parameter can take on. So I might find that a stress marker is actually indicative of relatively low frailty or low risk of mortality. This model thus allows me to examine or test aspects of the osteological paradox that were articulated by Wood et al. Over 20 years ago. And what those authors and others before them had argued is that we should not always assume that stress markers are indicative of poor health, even though that's how they're often used in bioarchaeology. And this argument rests on the fact that a lot of the stress markers that we use take time to form, so sometimes weeks or even months to form. And so an individual who has a stress marker had to have been minimally healthy enough to survive long enough for that stress marker to manifest, whereas somebody who lacks a stress marker might have been so robust that they were able to survive a stressor without a stress marker forming. Someone who doesn't have a stress marker might have been so frail that they succumbed to a stressor before that stress marker formed. So the real problem in bioarchaeology is the lack of a stress marker. Does that indicate really, really low frailty or very high frailty? So with this model, and in all the work that I do, I don't make any assumptions about what different variables indicate about underlying risks or about health in general. So I don't make any assumptions about what stress markers mean. If I'm looking at sex, I don't make any assumptions about which sex is at highest risk. I don't make any assumptions about whether high or low status is associated with higher risks of mortality. I use these kinds of models, and I allow the parameter estimates to indicate for a particular sample whether those variables are associated with higher or lower risks of mortality, and by inference about health. With this model, what I did was I applied it first to my normal mortality pre-Black Death sample to first establish whether the stress markers I was using were informative about elevated risks of mortality under normal conditions of mortality, and thus were potentially informative for looking at whether the Black Death might have been selective with respect to frailty. To examine other patterns of mortality, I used simpler models. So to look at the differences in risk between males and females, I estimated sex as a covariate affecting a baseline hazard of adult mortality, and to look at variation in risk with age, I simply estimated the parameters of the Seiler model. And for all of this work, I used maximum likelihood estimation, and specifically with a free program that's called MLE. So what I found in this previous research is that, contrary to long-held assumptions, the Black Death did not kill indiscriminately. And in particular, older adults and people of all ages who were in poor health, at least as suggested by the skeletal lesions, those individuals were more likely to die during the Black Death than their peers who were healthy or younger individuals. I did not find a strong effect of sex, but I do think that that might be an artifact of the way in which I modeled the sex covariate. So I do think that that's something that's deserving of additional study with larger samples. One of the things that I think is very important about these findings is that there was a general similarity in the patterns between the normal mortality sample and the Black Death sample. And so what that suggests to me is that we should always expect mortality to be selective, even under catastrophic conditions. So if the Black Death, one of the deadliest diseases in human history, if that disease was targeting individuals, we should expect that kind of selective mortality under other types of mortality circumstances. The work that I'm doing now examines whether this selective mortality during the Black Death, combined with rising standards of living following the epidemic, might have had substantial effects on health and demography in the post-Black Death population. So I have this consistent evidence that the Black Death was targeting frail individuals, and in particular it was targeting frail individuals of all ages, so not just older adults. And by doing so, and by killing people in such huge numbers, the Black Death might have exerted a very strong selective force on the population of Europe by weeding out the frailest, weakest individuals on a broad scale. We also have evidence from historical documents that in some places, standards of living improved, at least temporarily, following the Black Death. And that was a result of the massive depopulation that was caused by the Black Death. So there are fewer people following the Black Death to consume the resources that were around. And one of the things that has been observed in various regions following the Black Death is improvements in diet for everybody, and importantly, decreasing social inequities in diet following the Black Death. So everyone was eating more and better. And given that nutritional status can very strongly influence immune competence and health more generally, these improvements in diet for everyone could have had substantial effects on patterns of health following the Black Death. So what I've been doing is looking at how long people were living following the Black Death compared to a pre-Black Death condition. And I'm also looking at temporal trends in age-specific mortality rates. And my reasoning is, if the Black Death substantially affected health, and you're willing to accept my definition of health, which is not being dead, the longer that people can avoid being dead, the healthier they are in general. And that's a very, very unnuanced, basic definition of health, but it's incredibly useful for looking at samples of people who are dead. So if the Black Death and these changes in standard of living led to a population that was healthier in general, where people were, on average, having lower frailty, I expect that more people were better able to live longer in the generations following the Black Death than was true before the epidemic. I'm also looking at temporal trends in skeletal stress markers to look at patterns of health before and after the Black Death. And I'm going to focus today on the demographic results that I've obtained, but anyone who wants to talk to me later about some of the stress marker results, I'm happy to talk about those as well. So the project that I'm doing now will reveal the effects that the Black Death had specifically, and in London. It also, by inference, will give us more information about how emerging infectious diseases can affect populations in general. And it's also really important to understand what the post-Black Death population looked like in particular, because there were apparent dramatic changes in plague epidemiology over the very short term. So the Black Death, for example, as I said, killed an estimated 30 to 60% of affected populations. Some areas lost even more. The second outbreak of plague, which occurred in 1361, so 10 years later, killed about 10%, maybe 20% of affected populations. and that is a dramatic decline in mortality levels. So these aren't case fatality rates, these are overall proportions of populations being wiped out. And it's not clear why there would have been this decline in mortality levels following the Black Death, and so quickly. One possibility is that the Black Death led to improvements in health in general, and so people following the Black Death in general were less likely to die from a variety of causes, including, but not limited to, plague. Another possibility is that there were changes in susceptibility specifically to plague. So the Black Death wiped out most of those individuals in the population who were susceptible, and it took about 10 years to introduce new susceptible individuals via birth who could then be wiped out in the second epidemic. It's possible that there were changes in the population dynamics of non-human animal hosts for plague that altered human exposure to the disease in ways that affected mortality levels. It's possible that the causative pathogen, Yersinia pestis, changed in ways that rendered it less virulent. There could be other factors that I'm not considering. It could be a combination of factors. So the paleodemographic work that I'm doing now is part of a larger collaborative study that also includes genetic analyses of Yersinia pestis to see if there were changes over time that might explain the changes in virulence of Yersinia pestis. We're also looking at changes in human genetic variation over time to see if we are looking at genetic adaptation to this particular disease or changes in immunocompetence more generally. And we're also looking at disease ecology at the time of the Black Death, and I'll talk a little bit more about that later today. The project that I'm doing now focuses entirely on London cemeteries and cemeteries from within a relatively circumscribed area of London. And what I'm doing is I'm comparing pre- and post-Black Death assemblages from a single population to control as best as possible for population differences. I have 3 cemeteries that are providing a pre-Black Death sample. St. Nicholas Shambles and Guildhall Yard were both in use in the 11th and 12th centuries. St. Mary's Spittle was in use from 1120 until 1540, but it has 4 different phases of use. 2 of those phases predate the Black Death, so it's giving me pre-Black Death, and one of those phases from St. Mary's Spittle postdates the Black Death, so it's also giving me a post-Black Death sample. And then St. Mary Grace's is a cemetery that was established right when the Black Death ended, so it was in use from 1350 until 1538. So I have a pre- and post-Black Death from a bioarchaeological perspective, a fairly tight temporal range surrounding the Black Death, and also, as I mentioned, a relatively small geographic area. So I'm going to show you some of the demographic results that I've obtained, and I'll start with the results of Kaplan-Meier survival analysis. These are survivorship functions that show the proportions of people that survived to various ages. The pre-Black Death survival function is shown in blue, it's the lower one. The post-Black Death function is shown in green, it's higher. These survival functions are significantly different, and so this suggests that more people were surviving to later adult ages following the Black Death than was true before the Black Death. I've also looked at temporal trends in age-specific mortality rates, and the way that I did that was I pooled all of the age of death data from the pre- and post-Black Death samples to estimate a baseline hazard of mortality, and then I estimated burial in the post-Black Death period as a covariate affecting that baseline hazard. The estimated value of the parameter representing that post-Black Death covariate is negative. The 95% confidence interval for that parameter estimate includes just negative values. So what that suggests is that there were declines in age-specific mortality rates following the Black Death compared to the pre-Black Death condition. I did control for fertility. This is necessary in bioarchaeology because changes in fertility can affect age at death patterns in cemetery samples quite substantially, even if age-specific mortality remains constant. So we have this problem in bioarchaeology that we're looking at dead people. It would make sense to assume that what we're going to get from those dead people are mortality patterns. But in fact, a lot of the signals that we're generating are actually more informative about fertility than they are about mortality. And so I did have to control for fertility to make sure that these temporal trends I'm seeing in demography were actually telling me something about survival and mortality rather than telling me about fertility. So I used a proxy for birth rates, compared that proxy between the pre- and post-Black Death samples, and I found no significant difference in that proxy for birth rates, which suggests that the results I'm obtaining in terms of survivorship and mortality are not simply an artifact of changes in fertility. And for those of you who are interested in the specifics of that proxy, I'm happy to talk about it later in the question and answer period. So together, I'm finding consistent evidence that there were improvements in survival following the Black Death. There were improvements in mortality levels or mortality rates following the Black Death. And if we accept my definition that if you're not dead, you're relatively healthy, these suggest that there were improvements in health following the Black Death compared to the pre-Black Death condition. So, as is true of most research, this has just raised more questions than it has answered. And so some of the important questions that this has raised is whether the patterns I'm seeing are a result primarily of changes in diet, or a result primarily of changes in human genetic variation that might have led to a healthier population because of some underlying genetic variant. Was it a combination? Can I tease those things apart? Were the changes in demographics an artifact of changes in the patterns of migration into the city following the Black Death? So in the work that I'm doing going forward, I'm hoping to incorporate other approaches that will allow me to actually examine these different possibilities more clearly. In addition to the paleodemographic work that I do, I'm also involved in a collaborative project that includes molecular analyses of the pathogen that caused the Black Death, Yersinia pestis, which is shown in a micrograph here. We're also looking at the disease ecology at the time of the Black Death, and also looking at human genetic variation across the pre to post Black Death period. And this is me wearing a clean suit to obtain samples for this ancient DNA work. So I obtained samples of bone and teeth from people who died during the Black Death and were buried in East Smithfield. And the team that I'm working with has generated the first complete draft genome of 14th century Yersinia pestis. And this is really important because ancient DNA analyses going back to the 1990s had produced some evidence, some of it was highly debated, but some evidence that the Black Death was caused by Yersinia pestis. But none of the previous ancient DNA analyses had or were able to address epidemiological changes in that pathogen. So for example, were there genetic reasons why the medieval plague became less deadly over time? We also have evidence that the Black Death was capable of being spread human to human much more easily than is true for modern bubonic plague. So now, because we have the full draft genome of Yersinia pestis from 14th century Yersinia pestis, we have the potential now to try to find molecular reasons for these changes in epidemiology. Comparison of the 14th century strain of Yersinia pestis to a modern reference strain has not yet revealed any significant functional differences between the two that can explain why the Black Death was as deadly as it was. So far, this work has not yet revealed the smoking gun that allows us to pinpoint these genetic reasons for changes in plague epidemiology. This is, however, an ongoing project. We're using additional samples to make sure that we haven't just missed a key difference in that initial comparison. We're also looking at changes in human genetic variation by comparing samples of individuals from the pre-Black Death assemblages to those from the post-Black Death assemblages to see if we can identify any changes in human genetic variation that can be tied to the Black Death. And then we're also looking at the possibility of a syndemic at the time of the Black Death. So there's this possibility that the Black Death might have been as deadly as it was because there were multiple infecting pathogens at the time of the epidemic. And what can happen with infections with multiple pathogens is within host competition that can drive up the virulence of those infecting pathogens. So what we're doing is we're starting to look at what was circulating at the time of the Black Death, and can we possibly identify signatures of this type of syndemic during the Black Death. So one of the things that has come out of this collaborative project so far, using one individual from East Smithfield, is a microbial detection array that has identified thousands of environmental, microbiotic, and pathogenic taxa, at least at the order of the level. So this does mean we have this potential to really look at the disease ecology at the time of the Black Death, and potentially to identify things that might have been competing with Yersinia pestis that might have driven up the virulence of Yersinia pestis. However, the possibility remains that at the end of the day, we might never find a significant difference, a significant functional difference between 14th century Yersinia pestis and modern Yersinia pestis. We might not find a significant difference in the disease ecology at the time of the Black Death compared to modern bubonic plague that would explain the very deadliness of the Black Death. We might not find substantial changes in human genetic variation between the pre- and post-Black Death periods. And what we might find is that just as with the current Ebola epidemic, that it's the human context of the disease that's crucially important for determining many of the characteristics of the disease. And so it's very important to keep our focus expanded beyond just the pathogen itself and also consider the human context at the time of the Black Death. So just briefly, some of the work that I'll be doing in the future is going to examine what might have favored the emergence of the Black Death in the 14th century. I'm particularly interested in famine. There were repeated famines in the pre-Black Death period, and given that nutritional status affects health and immune competence specifically, those repeated famines might have lowered health or increased frailty, or at least heterogeneity and frailty in the pre-Black Death population in ways that made the population particularly vulnerable to the Black Death at that time. So far, what I've been doing is looking at demographic trends between two pre-Black Death periods, so from 1000 to 1200 AD and 1200 to 1300 AD. And so far, from the skeletal data, I'm finding substantial decreases in survivorship in the 13th century, so just before the Black Death, and increases in mortality rates, again in the 13th century, so just before the Black Death. Starting this summer, I'm going to be collecting data from individuals who are buried in purported famine mass burials. And I hope in the near future to incorporate isotopic signatures of starvation to look at patterns of variation and frailty under conditions of famine mortality, and to look at health in general in the context of these famines in the pre-Black Death period. I'm also very interested in the health of migrants before and after the Black Death. As I mentioned, the results that I'm finding in terms of the demographic trends following the Black Death might have been affected by migration patterns after the Black Death. And so what I'd like to do in the near future is examine migration patterns before and after the Black Death and test the migrant selectivity hypothesis that migrants into London were a select subpopulation of healthy individuals versus the idea that migrants are particularly vulnerable to diseases once they migrate into cities, and particularly to diseases that are endemic in the cities that they did not experience during childhood. So I'll end by emphasizing the ways in which this kind of research on epidemics in the past might produce benefits that are applicable to living populations. So research on disease in the past can reveal the ways in which people vary in their susceptibility to disease and death during disease epidemics. So for example, I have these results from the Black Death indicating that people who were suffering from disease or malnutrition before the Black Death were therefore more at risk during the Black Death than people who were relatively healthy. And as I mentioned before, I found general similarities between the normal mortality sample and Black Death sample, which suggests that we should always expect mortality to be selective under all conditions of mortality. And I think that should encourage us to identify the ways in which people are going to vary in their susceptibility to disease and death in living populations so that we can actually do something to reduce those risks so that we can reduce overall mortality levels in outbreaks in the future. Evidence of short and long-term demographic and health trends following epidemics can really allow us to understand the power that epidemic diseases have to shape populations, and also the role that human demography can play in terms of driving the dynamics of the disease and also the evolution of the disease. And this evidence can inform our models of the evolution of virulence in emerging pathogens and help us to resolve questions about whether changes in emerging disease epidemiology are the result of human adaptation to the disease, or changes in human demography or behavior, or evolution of the causative pathogens, or some combinations thereof. Improved understanding of these issues can enhance our abilities to to deal with emerging diseases in living populations. So for example, we can potentially reveal which pathogen or host factors could feasibly be altered in order to reduce risks in living populations. So if we find consistent evidence that it's population density that has the strongest effect on disease virulence, that's perhaps less easy to deal with than if we find consistent evidence that it's the nutritional status of affected populations that can really drive mortality levels. That's maybe not easy to deal with, but easier than dealing with population density. Also, pathogen genomic data can allow for the development of drug therapies and vaccines. And this has happened with current diseases. So, for example, a drug was developed using pathogenic orthopoxy virus genome data in order to treat a child who developed life-threatening complications from a smallpox vaccine. So very quick development of that drug. The genomic data from various pathogens has revealed vaccine antigens. So with research on disease in the past, if we can apply these ancient biomolecule approaches and add to existing genomic databases, that might provide public health benefits if virulent strains of diseases were to reemerge in the future. And lastly, I think that ancient disease research can really be used to harness and raise public awareness about the powerful effects that diseases have on our species. And I think that the Black Death is particularly useful in this respect because everybody is interested in the Black Death. And I don't think it's just people are being polite because they know I study the Black Death. I think it really just fascinates people. So you see it referred to in novels. I'm sure many of you have read A Year of Wonder, Doomsday Book by Connie Willis. It's in movies. There was a Nicolas Cage movie. Monty Python, The Holy Grail. There are Black Death cigarettes, Black Death vodka, Black Death beer. People wear Black Death tour t-shirts that have the dates at which the Black Death appeared in various cities. People have Black Death tattoos. Giant Microbes makes a stuffed Yersinia pestis toy called the Black Death. So it's everywhere. People are fascinated by it. And I think that we can use the Black Death not to generate fear at all. I don't want to do that, but I want to tap into people's inherent interest in this particular disease in order to motivate action in the face of threats to living populations. So thank you all for coming today, and just like to acknowledge people and funding agencies for this work. Yes?
We have about 10 minutes for questions.
Okay.
Two quick questions.
Yeah.
One is we had a variety of frailty factors.
Mm-hmm.
Are some of them more risk factors than others? So there's selection for particular kinds of frailty?
So in terms of like the stress markers that I've been using, so let me show you some of the stress markers. So some of the stress markers that I'm using are indicative just of stress that occurred during childhood. I'm not showing it. I'm showing it with myself. Short adult stature. So that's something that's revealing developmental stress. That can be the result of disease or malnutrition during childhood. Crib or orbitalia. And product hyperostosis or porous lesions that form on the orbital root or the top of the skull, usually attributed to anemia but can be caused by other things, but importantly only develop during childhood. So those are childhood stressors. Enamel hypoplasias, interruptions of enamel formation during childhood. I also do look at periosteal lesions, which is abnormal proliferation of bone that can occur in response to trauma, localized or systemic infection at any age. So a lot of the things are indicating developmental stress, and some are indicating stressors that can occur at any time. Going forward, it would be possible to apply stable isotopes to look more carefully at some dietary differences that might tell us something about diet in adulthood as opposed to just childhood. So I can't tell at this point whether I'm looking at nutritional stress, disease stress.
But if you use any of these in a univariate model or something, and just look at them, are some of them more predictive than others?
I'm not comfortable saying that because, well, let me show you more stuff. Okay, there, this is a good one. So I mentioned previously that I used my pre-Black Death sample to examine differences in risk between those with and without stress markers. So this, for example, is saying individuals who have proliferative lesions on the tibia are 430% more likely to die than their age peers who don't have them. I would never actually report individuals are 430% more likely to die with these lesions than their peers without them, but it does show a general trend. These are the Black Death patterns. Again, elevated risks. They're lower than those in the normal mortality sample, which might indicate reduced selection during the Black Death compared to normal mortality, or it might be just an artifact of sample composition. But one of the things that this has suggested is that there were people who were healthy during the Black Death that died that might not have been likely to under normal conditions. That reduced the difference in risk between those with and without. But the sort of the numbers, specific numbers I'm getting, I don't actually pay that much attention to them. I just look at general patterns. So, yeah.
The related question actually you started to address, which is further you can use fingernails or toenails as a temporal record of nutrients.
I'm sorry, what?
Of nutrients of what people ate in the last month. Right?
So with skeletal studies, we're looking at teeth and bone, and you can.
No more fingernails or toenails.
Not in the stuff that I'm using generally, thank God, because I don't want to look at that. But you can look at childhood versus more recent, if you're looking at adults, childhood diet, if you look at incremental dental, if you take samples. So you can look at change over time in diet within a single individual. Mm-hmm. Yeah.
So my first question has to do with the sample size,
Mm-hmm.
the sample composition.
Yeah.
Speak up and repeat the
Yes.
question.
So the question is about sample size and sample
I'm just
composition.
wondering about your sample size.
Yes. So for my previous research, I had a sample of 491 individuals from the East Smithfield Black Death Cemetery and about 400 or 300 individuals who made up my pre-Black Death sample. Basically, if you're using a hazard model with about 3 parameters, you want to have a sample of at least 50 to 100. The more parameters you add, the much higher, larger your sample has to be. In terms of composition, it's not entirely clear what the composition is. East Smithfield, okay, so East Smithfield had both mass burial trenches and also individual graves. And you can see the single interments here and then part of a mass burial trench there. Some people have argued that those trenches were used for lower status individuals, individual graves for higher status. Other people have said, well, it might be that the mass burial trenches were used at the height of mortality when there was less time to dig individual graves. There's no temporal information within the cemetery to tease those apart. People are buried in standard medieval Christian fashion, no grave goods, so we can't get at status at this time period. So I do know that I have all ages, both sexes, but in terms of other things, occupation, status, I can't get from the skeletal data. I can get, though, from collaborating with historians who have access to records that talk about occupation and status, but there are limitations to these data.
And then the second question has to do with the culture.
So
you started to touch on. I'm just wondering, in the historical records, can you look for, patterns of behavioral change as a function of, post Black Death, or housing or anything having to do that? In addition to, when I look at the maps of the Black Death in Europe, I've always been intrigued that there are pockets
where
like
it didn't.
in Finland where there's just zero.
Right.
And so, what is going on culturally?
So something that did happen following the Black Death in certain areas is the quarantine measures were enacted in order to try to prevent the spread of the disease. In terms of what's happening during the Black Death, the places that apparently escaped it was probably because you didn't have people entering those areas to introduce it, if it really truly was spread person to person. But yeah, there's a special issue of the Medieval Globe that I recommend reading. There's a lot of information that's been produced by historians, not just in Europe, but outside, that might address some of those questions.
Open access.
Open access. Yeah, Melissa.
Yeah, I was curious. You said that there wasn't a sex difference, but
Yeah.
that you thought there might be.
There-
I
well-
wanted you to talk about it, and
Yes.
also with the ancient DNA, you're looking at the pathogen, not the human
Not
yet.
Well, nothing published with human data yet. So yeah, so that's a work in progress, right? So far the ancient DNA has focused on the pathogen. Okay, so your first question was, why am I questioning the results I found for the sex difference? There's so much evidence to suggest that we should be finding a difference in sex patterns of mortality at the time of the Black Death, because it's a human universal now that females are just better than males. I mean, let's just face it, for a variety of reasons, but hormonal reasons, behavioral reasons, chromosomal reasons. Males are much more vulnerable. If you look at all populations for which we have data, there are lower age-specific mortality rates for females and longer lifespan for females. Where we find exceptions to that, they're usually explained in cultural terms. Anne?
Are pregnant women though more
This is what I'm going to get to, yeah. So in general, females are less frail than males. The exception is that we tend to find higher risks of females during the reproductive ages. So pregnancy can put females at higher risk of certain maternal mortality, certainly. Men don't suffer from that. But also there are diseases like malaria, which produces much higher mortality in pregnant women, particularly women in their first pregnancy, than is true for males of the same age. So what I did when I modeled the sex covariate was I used a proportional hazard specification that doesn't allow for variation in the difference in risk of death between males and females across age. So it's possible that the lack of difference that I found is an artifact of sort of this averaging effect. So maybe females suffered lower risks of mortality at very young ages and at very old ages, but at the peak of reproductive ages, they might have suffered higher mortality during the Black Death than males did. But by looking at all ages simultaneously, I'm unable to detect that. So that's something that I can't actually add parameters to that to look at that variation without using a larger sample size. So people need to get out and dig up Black Death victims and then send them to me so I can actually look at that. And then your second question, the human genetic variation is key. If we're actually going to look at human pathogen coevolution, we have to look at the evolution of both the human and the pathogen. And we're starting to look at the pathogen and not finding anything, and it could be that all the answers lie within the human body, the human genome, the human population. Yeah, Monica.
Your samples are obviously all coming from Northern Europe, so you've also used, is it Danish samples?
Danish samples, yeah.
Samples as well as you know, the evidence we currently have suggests that there were a lot of places that were affected by the Black Death that didn't recover. That there were very long-term demographic.
And that's true for England as well.
Yes. And so I was just wondering how. Well, let me put a hypothetical on. Let's say that somebody just turned up a cemetery in Cairo. What kinds of things similarly could you do with something that's in a really different part of the world? Or China, which is obviously what we're going to be looking at.
So if I had access to those sorts of cemetery samples, I would apply the same approaches to look at and see if these patterns are consistent. So are we seeing the same sort of selectivity? Was there variation in the strength of selectivity during these epidemics? I mean, you highlighted an important point. The results that I'm getting are specific as far, I can't say anything more than London. And that's true for a lot of the historical documents. They're very rich for some areas and very, very poor, non-existent for others. So the first step would be, am I seeing consistent patterns between London and Cairo, London and Pisa, and Genoa, and Marseille, and all these different places? And I think that there are limitations to what can be said with the demographic data, but if we could pull together demographic data across different regions of the old world with the human genetic data, with the pathogen data, that would allow us to actually have the power to potentially tease apart what was really happening following the Black Death. Was it diet? Was it human genetics? Was it the pathogen evolving? And how might that have varied based on environmental conditions, social conditions, economic conditions? These increases in standard of living were not consistent, and they were not uniform across even Europe. So that's something, those improvements that I'm seeing in London might not also have been happening elsewhere. So that'd be another question, is what were the short and long-term effects after the Black Death, not just patterns during the Black Death? So again, get out, dig up Black Death bodies, send them to me.
On that note, I believe that our time is up. If anybody has any further burning questions or wants to chat. We have a little time late this afternoon, so you could email me and I'd try to arrange something. Okay?
Thank you.
Thank you.